sorry, in, yes, in violet, sorry, you have a maximum of 1,645. Okay, so it's probably something here under red conformation. And you have also a shoulder in this band. You have a maximum here and a shoulder in this band, which correspond also to the maximum of a small sequence of these proteins from 27 to 30, a maximum at 1,630 centimeter minus 1, which corresponds to the beta sheet. Okay, so the healthy one has alpha helix, and the pathogenic one has beta sheet and also underred conformation. Is it okay for this first question for infrared spectrum? Yes? Okay. So let's go after to the CD, circular decrease spectrum. We measure a CD spectrum of one of these proteins. So you have three proteins, one with alpha helix, one with beta sheet, and the other one with a random core. Okay, and record the CD spectrum. You observe two minimum around 200 nanometer, which correspond to the alpha helix. So if we know that this protein, this spectrum belongs to PRPC. Is that okay? So if you yes, if you see the slide from the prion here, so you have the healthy proteins with several alpha helix, and when it becomes pathogenic, you will have alpha helix, but also two alpha helix which change in their conformation and becomes beta sheet. And because you have beta sheet, you can have interaction, hydrophobic interaction between the beta sheet of several pathogenic prion. So it causes dimerization and then aggregation. So you will have the amyloid, which is responsible for the neurodegenerative disease. Okay? Do you have another question? So, no, I can start now the last part of lecture about proteins. So we've already seen the primary structure, which corresponds to the sequence of amino acid in the proteins. The secondary structure, which corresponds to the local structure of one part of the proteins. So you have three major secondary structure: the alpha helix, the beta sheet, the beta term, and also this is the three major secondary structure. Okay, so now we have the third tertiary structure. Okay, so the third type of structure found in proteins, it's called tertiary protein structure. And it will give the shape, the 3D structure of the proteins. And all the side chain of the amino acid will have interaction with other amino acid. So all this weak interaction will give the 3D structure of the proteins. So now you know that several types of interaction between the side chain of the amino acid. So the stronger one, which are the stronger one? Which is the stronger interaction in all this example? You have disulfide bond, ionic bond, hydrophobic bond, hydrogen bond. Which is the stronger one?
The disulfide bond?
Yes, correct, because it's a covalent bond. So it's a stronger interaction. And how can we break this disulfide bond? If we want to break this bond, how can we do that? We can use what type of chemical molecules, chemical agent, chemical reactant to break this bond?
I think we can use the reducing agents.
Yes.
Like beta mercaptoethanol.
Yes, correct. Beta mercaptoethanol. So it's a reducing agent, and he will break the disulfide bond. Perfect. After that, the ionic bonds, for example, between lysine and so the positively charged of the side chain of lysine and the negatively charged of glutamate, for example. After that, you have the hydrogen bond. You can have hydrogen bond between the peptide bond, so the skeleton of the chain, but also between the side chain of the amino acid, for example, serine, and the CO of the peptide bond, but also serine with the side chain of glutamate. Remember you that for an alpha helix, you have also hydrogen bond, which allow to form the alpha helix. And finally, the weakest interaction will be the hydrophobic interaction between hydrophobic side chain. You have also what we call p-stacking interaction. It's like, it's here. It's already here. So the p-stacking interaction, it's like a hydrophobic interaction and van der Waals interaction. It's the same kind of interaction, but the p-stacking, it's because you have aromatic ring which stack together, and you can have interaction between them, and we p-stacking. We can observe this kind of interaction between aromatic amino acid in the protein structure, but the most important P interaction, it will be in the DNA and RNA. Okay, you can have the of DNA or RNA which stuck together, and form the helix of the DNA. You know that. Okay? So this is the same interaction. Okay. So we can we can mention what is the denaturation and renaturation. So here you have the 3D of a normal proteins. So it's called the fold proteins. And when this protein is submitted to an external stress, like or external compound, like the temperature, when we increase the ionic strength, when we increase the salt concentration, or we add organic compound, for example, the 3D protein will be unfolded. So it's what we call the denaturate proteins. So the weak interaction will be broken because of the external stress, and so we obtain only the the only structure that remains when we have the denaturate proteins. It's only the primary structure. It means that when you have stress, like temperature of the presence of alcohol and so on, you will break the weak interaction, but not the peptide bond. Okay, so you will have only the sequence of your proteins. So denaturation involves the destruction of secondary and tertiary structure, but not the primary structure. Okay. And so the most common observation in the denaturation process of a protein will be the precipitation or coagulation of the proteins. Okay, so sometimes you can observe the precipitation of the protein, precipitation or coagulation or aggregation of the proteins. And we will see in an example, we try to avoid these denaturate proteins, for example, in the pharmaceutical industry. I will show you an example. So, what is important? So when you have denaturation and when you can avoid the precipitation, we can have the reverse reaction. You can have the renaturation. It means that you will have the unfold proteins. And when you put these unfold proteins in its physiological condition, in the same condition, you can return to the normal protein, the fold protein state. Okay? So the denaturation and renaturation can be a reversible process. Okay? So the first stress, external stress, so it can be the thermic denaturation. It means that you will heat your proteins and you can disrupt hydrogen bond and also hydrophobic interaction when you heat the solution. Okay? And so we can measure what we call melting temperature of a proteins. The melting temperature It's the temperature when 50% of your protein are fold and 50% are unfold. It's like when you measure the melting temperature of DNA, okay? You can have the same definition. It's the temperature at which 50% of your protein is denaturate and 50% of your proteins are in its 3D structure. Okay. Can you give me some example in the day life when we can observe the denaturation of protein with the heat?
When we boil the egg,
Sorry,
when we boil the egg,
I don't understand.
When we cook
Yes, yes,
the egg,
yes. Perfect. Yes. So when we cook, you will break all the band, and for example, in the eggs, it's become you will denaturate the protein of the eggs, and it will coagulate during the cooking. So that's why you have the eggs, the white part of the egg. It's become from transparent to white. Okay. And other food also are cooked to denaturate the protein to make it easier for the enzyme to digest them. Okay. And a second example, you have another example, maybe in medical application, when you want to sterilize the instrument that we will use for medical surgery, you will sterilize the instrument by heating them. So you will denaturate the proteins in the bacteria or virus, for example, and destroy the microorganism, bacteria, virus sometime, and so on. Okay. For example, in the lab, to avoid the presence of bacteria, we probably use autoclave. To sterilize the buffer, and then you can use them as physiological media without any bacteria. Okay. So for the chemical denaturation, for example, you can have alcohol, so the alcohol, which kind of bond will alcohol like? No, I didn't hear your answer.
I think it's a hydrogen point.
Yes, indeed, because alcohol, so you know, ethanol have an OH group. So this OH group can form hydrogen bond. So you will disrupt H bond. Okay, so all the H bond in your protein will be disrupted by the addition of alcohol, and that's why we use hydroalcoholic gel. Now we know that with the pandemic, with the COVID, we use hydroalcoholic gel to wash our hands. Okay, so normally you can use a solution of 70% of alcohol. So at this concentration, the alcohol is able to penetrate the bacterial of the virus cell wall, and so denaturate the protein and the enzyme inside the microorganism. If you use a higher concentration of alcohol, so if, for example, you use a pure alcohol, 95% of alcohol, at this concentration, the alcohol will coagulate the protein outside the cell. And so he cannot disrupt the protein, the bacteria of the virus wall. Understood? Another chemical denaturation, it will be acid or base. So what kind of interaction will disrupt acid or base? Or when we change the pH of the solution, what kind of bond, weak bond, can we break?
The ionic bond.
Yes, ionic bond. So when an example, it's in your stomach, for example, in your stomach, the pH is very low, around 1. So it will denaturate what you eat, the protein you eat. And that will becomes easier to digest the food. Okay, so it's acidic gastric liquids in your stomach. We can which ease the digestion of your food. You can have also heavy metals. So, for example, mercury, lead, silver, and so on. So because they are salt, they will disrupt also the ionic bonds. And you can have also, we will see on the last lecture, we will see that these metals can interact with the side chain of the amino acid and form what we call a complex. So this is used, for example, the silver is used as a disinfectant. Okay, so it can be used to prevent infection in the eyes, for example, or the treatment of nose and throat infection. Mercury salt, I don't know, in Vietnam it's the same name, but mercurochrome. We use This is the name in Vietnamese. No, I don't know. Okay. Okay. So this mercurochrome we use in France, and they have they have similar properties. They have properties in preventing infection in wood. Okay, but the same reaction, it means the bonding of the heavy metal to proteins. Its value can be used in case of empoisonment. It means when you have a very high quantity of heavy metal, one of the antidote is to heat a lot of proteins like milk or eggs, and then you will all the metals you eat will precipitate with the proteins. Okay? And so the precipitate metal proteins is then discharged from the body. So you have here a summary of all the stress we can have for the proteins. So the temperature, I already told you that it will modify the H bond and the hydrophobic interaction. The pH, it means the acid and base addition modifies the ionic interaction. You can have also the salt concentration of your solution. It will be increased, modified the ionic interaction by competition. Detergent, organic solvent, and urine, it will modify hydrophobic interaction. So detergent, we already see that, but in lab, we use SDS, for example. You remember the SDS, it's used when you want to denaturate the protein before make the SDS electrophoresis. And the SDS has a hydrophobic tail which interact with the proteins and disrupt the hydrophobic interaction in the proteins and denaturate the protein. In the organic solvent, so we see alcohol, so it will disrupt H bond. But also if you use apolar solvent, for example, DMSO, you will disrupt the hydrophobic interaction. So that's why usually the drugs are hydrophobic, and to dissolve the drug, we use an organic solvent, usually DMSO or alcohol, but the percentage of DMSO of alcohol should be low when we study the interaction between the drugs and the protein, because if you use a high concentration of DMSO of alcohol, you will disrupt the protein and then denaturate the proteins. Okay? Urea, so it's this molecule. He can disrupt the hydrophobic interaction and also the H bond because of the presence of the oxygen and nitrogen. And we have already see that, but the redox region or reducing region like mercaptoethanol or DTT will break the disulfide bond. Okay. So techniques to study the denaturation is this technique. So it's a differential scanning calorimetry. So it's the most direct and sensitive approach to characterize the thermodynamic parameter which control the weak interaction in the proteins. You in this experiment, it's require only few milligrams of proteins. So we will, maybe I will start by this. How to perform a measurement with DSC. So you have here two cells in adiabatic shield. In one reference, you can put the buffer. In the sample cell, you will put your proteins. And you will rate, both cells are hit at a constant scan rate. It means that the temperature will increase slowly and in the same time. And the absorption of heat that occurs when a protein unfolds, because you will break the band, will cause a temperature difference between the cell and resulting in a thermal gradient across this adiabatic. So we will measure what we call the heat capacity against, the heat capacity against the temperature. So you will heat this, and so we will observe this kind of curve. And just remind that we can obtain from this curve the melting temperature. It means the temperature at which 50% of the proteins are denatured, 50% of the proteins are old, native or denatured. And you can have the heat enthalpy of the fold. Okay, for us, we will see that the higher the thermal transition, so the melting temperature, the more stable the molecules. Okay, so remind this: the higher the melting temperature is, the more stable is the proteins. Okay. So these techniques, the DSC, cannot give any information about the tertiary structure, but will give you the melting temperature. Okay, so in this example, this author used two proteins, Barnase proteins and ubiquitin proteins, and they perform the DSC experiment at different pH value. And here you can see that at pH 5.5, the protein is more stable than at pH 1.8, because the melting temperature is higher at pH 5.5. And in the same way, in the same case, you have the ubiquitin, which is more stable at pH 4 than at pH 2. The other techniques to obtain those tertiary structure now, it means the position of all the atom in the protein, the 3D structure. So you have the X-ray diffraction. So it's a process with high cost, with high cost and very long treatment of data. The limit, because can be a process can be highly cost, can be expensive. Sorry, I have to find my words. So this process can be expensive because you need a lot of proteins. So you need to purify the proteins. It can be very time and you can. Sorry, the purification of the protein can take a lot of times, and also you have to crystallize the proteins. The limits, it's for membrane proteins, but for now we have techniques now to obtain crystal for proteins, membrane proteins, so it's okay. So the X-ray diffraction, it means that you have the 3D structure of a protein in a solid state. So not in the physiological state, because maybe when the proteins become solid as a solid, it can change its conformation and the 3D. So another way is to use nuclear magnetic resonance, NMR. But so in this experiment, you are in liquids, in solution, so you can use physiological condition of your proteins. But the limit, so you should work on small proteins because you have a very long treatment of data. So if you can have X-ray diffraction or NMR, you can use molecular modeling. Okay, so it's theoretical chemistry, computational techniques, and we will, because you have a lot of similarity or identity isn't in the sequence of proteins, and we have a lot of data, we can guess the structure of the protein because of the high homology of the proteins in the same family, for example. So here you can have an example when you have the superposition of 3D structure determined by NMR, so in solution, and by X-ray, so it means as a crystal. So you can see it's almost similar, except in this area, it's the random call. It's not structured, it's not ordered. And you have also the position of this helix, which is different in the crystal than in the solution. Okay, so be careful when you have a 3D structure. You have to be aware by the techniques used to determine the 3D structure. So, usually proteins are described as several structural units. So, its structural domain is an element of the proteins of a whole structure, and that is self-stabilized and often fold independently of the rest of the protein chain. Okay, so in your proteins, you can have several structural domains which can be folded independently. Okay, and many domains are not unique to the protein produced of one gene, but instead appear in a variety of proteins. So you can find this structural domain in several proteins. And the domain are named because of their biological function, for example, or their secondary structure or biological function. Sometimes we can call a domain the calcium binding domain of calmodulin because it's very specific of the calmodulin, but we can find this domain in another proteins. Okay? So I'll give you some example of a structural domain. So you have the alpha made by alpha helix, alpha domain. This we will see when we study the membrane proteins. It's a beta barrier, so composed with beta sheet. You have also hemoglobin composed with two beta domains, this one and this one. The bacteriochlorophyll made with the beta sheet and also alpha. So, oh, this is the same. We will see also that in the membrane protein. So you have the alpha beta domain team barrier. So it's like a channel. We will see it's very important. And so on. Okay. So let's see. Yes. So I want to show you an example of the use of DSC, differential scanning calorimetry. So it's used in pharmaceutical industry to know the stability of your protein in the presence of different excipient. Okay. So in this example, the author used human serum albumin, HSA, Y, and drugs, which is called SEPFB4. Okay. So the novel technology are emerging to produce therapeutic drugs, but we have to enhance their pharmacokinetics. And in order to do that, they usually construct a fusion protein with a drug with another protein to enhance the circulation time in vivo. The protein used, the fusion protein used, usually are antibody fragment or the human serum albumin. Okay, and why we use the human serum albumin as a therapeutic drugs candidate? It's because you have this protein in high concentration. So it's between 30 and 50 milligram per milliliter. The HSA has a high half-life, of 90 days and has restricted biological activity. So we can use them as a carrier molecule or transporter molecule as you want. Okay. And also, maybe they guess that the fusion protein with the albumin can cross the blood-brain barrier. P. Okay? So the drugs, you can interrupt me if you have questions. The drug, indeed, the EPSB4, these proteins, can interact with the F3B2, okay? So the EPSB4 is a member of the kinase receptor. And when this interact with F3B2, he induces a signal and will play a key role in the vascular development. And the EPSB4 is induced in a lot of cancer, like breast cancer, head, neck cancer, colon cancer, uterus cancer. Okay. And when you have a high concentration of EPSB4, it means that it predicts a higher state of cancer. So an effective antagonist of this is the soluble part of this receptor. So it's called SEPLB4. It's only the soluble part of this protein. And this soluble part, so the smaller proteins, can interact with F3B2, and so it will inhibit the tumor growth of in some models. Okay. So it's a promising therapeutic candidate, this, for the treatment of cancer. And then to improve the utility of these drugs as a drug candidate, they design a fusion protein between this drug and HSA. And that they will study the stability of the fusion proteins in the presence of excipient. So in this graph, you can see the DSC experiment. DSC experiment of the fusion proteins. What we observe. We observe a different transition. 1, 2, 3. We have the first here, a second one here, and another one here at the shoulder. Okay. So we have a transition at around 7 degrees. Another transition at 68 degrees Celsius. And the last one, the shoulder one, the rod at above 70 degrees. Okay. And indeed, this transition corresponds, if we compare this graph to the denaturation experiment, the DSC experiment of the proteins alone for the SEPF before, the DSC experiment show only one transition at 57 degrees. And for the HSA, you have two transitions at 68 and more than 70 degrees, corresponding probably to the dimerization of these proteins. It means that the fusion proteins have a behavior like it's different. It's like two individual proteins, okay? They are this to these proteins, but all the structure of each protein stay similar to the protein alone. And so these fusion proteins act like it has two proteins, okay? A structural domain. I told you when you have different structural domain, the structural domain in proteins can fold independently, so can be also unfold independently of the other protein. And this is the case here of the two proteins which are fusioned. Understood?
Yes.
Yes? Okay. Thank you. So you can have in this table the effect of excipient on the thermal melting temperature. So the TM1 corresponds to the SEPF before, as the TM2 corresponds to the melting temperature of HAs, the albumin. Okay? So in the presence of sugar, different carbohydrate, you have what we obtain, a stability of the albumin because the TM increased, and also a stability of the SEPH4 because the TM1 is higher than in the control. Okay. In the presence of CD, so you know what is the CD? It's cyclodextrin. CD. This is this molecule, the cyclodextrin. It's a ring composed by sugar, so usually glucose. Okay, you have, depending on the number of sugar, you have alpha, beta, and gamma. And what happened? You have inside an hydrophobic cavity, and outside is hydrophilic. Okay, and I already told you that usually the drugs are hydrophobic. So to improve its circulation or improve the solubility of these drugs in blood, for example, we use cyclodextrin, and then the drugs can interact in the cyclodextrin, and so you avoid the interaction of the hydrophobic of the drug with water, okay? Because you have hydrophobic interaction with the cyclodextrin, and outside you have hydrophilic polar group. Anyway, the presence of the cyclodextrin did not improve the TM1, and also it destabilized this fusion protein, because you can see that the TM2 value are lower than in the control. Okay, and the third type of excipient is the salt. And you can see that in the salt with high concentration, we have the same TM1 and TM2. Otherwise, in the case when you don't have any salt, you have degrees. So sorry, I will give by increasing the concentration of salt, you increase the stability of the fusion protein, the albumin part. Okay. So here you have the fluorescence, tryptophan fluorescence intensity of the fusion protein at different pH. Okay. When we said tryptophan fluorescence, it means we excited at which wavelength value? What is the excitation wavelength?
280.
Yes, 280 nanometer. Okay, and we measure the intensity of this fluorescence against the concentration, and it's okay. We observe a degrees of this fluorescence intensity, because when you increase the temperature, you will increase the non-radiation decay. Now we plot the center, the maximum indeed, of the fluorescence against the temperature. It means that when the maximum increase, you had a red shift, and when he decrease, you have a blue shift. And we already see that when we increase, maybe not, I don't remember, we will see that. When we increase the temperature, usually we observe a red shift. This is the case of the black plot at pH 3. Remember why we increase the temperature? We observe a redshift. Maybe I can explain again. So, sorry?
I think it's because the protein is the unfold. So neither the ionic or the solvent exchange, so it's caused the redshift.
Indeed, remember that we excited the tryptophan. Tryptophan is a hydrophobic residue, so usually it's inside the proteins. When you increase the temperature, you will unfold the proteins, so the tryptophan become surrounded by water because you will have the denaturation of your proteins. And you should know that the fluorescence is very sensitive to the polarity of the solvent and the polarity of the tryptophan environment. And when the polarity of the tryptophan environment increase, it means when the tryptophan is surrounded by more and more water, you observe a redshift. Okay? But at pH 5, what we observe a blue shift at 50, 60 degrees. What happened? What happened here? If you have the blue chief, it means that you have aggregation of your protein. Okay, and this aggregation, this coagulation of the proteins is something the pharmaceutical industry try to avoid. Okay, the aggregation is one of the major physical degradation for proteins in pharmaceutical industry. Okay, and this is the most important thing they try to avoid. So it's very important to avoid this aggregation, because when you have the redshift, it's only a denaturation, and you know that this denaturation by temperature will be reversible. So when the temperature becomes lower, you will normally have your native proteins, but not when you have aggregation. Okay, so then try to observe the aggregation of the fusion proteins in the presence. They try to avoid this, so they perform the experiment at different concentration of sugar in the presence of sugar at different concentration of salt, and with sugar and salt. Okay, so when the concentration of salt is 0, you have an aggregation temperature of around 55 degrees, but when you increase this concentration, the protein become more stable because the aggregation temperature it's around 70. Okay. The nature of sugar has no effect on the temperature of aggregation. And so in this case, we see that the best condition to store these fusion proteins will be 150 millimolar of chloride sodium in the presence of 10% of sucrose, because you will have the aggregation temperature around 75 degrees. Okay. We will do the exercise after the coffee break, but I will finish the lecture. So it's the last part of my lecture, and this concerns the quaternary structure. So the quaternary structure is the arrangement of multiple fold protein. So when polypeptide change, when in protein you have several polypeptide change, this change can assemble into multi-subunit structure, and the arrangement of proteins or protein, what we call subunits, in 3 dimensions constitutes the quaternary structure. Okay, so in this example, you have a homodimeric proteins. It means that you have 2 chains which are identical. You can have heterotetrameric proteins. It means that you have 4 polypeptide chains, 4 subunits, but which are different to each other. So in not next week, but the week after, we will see myoglobin and hemoglobin, and we will see that the quaternary structure of hemoglobin is very important. Okay, so I would give you some example. You have dimer, trimer, tetramer, and so on. And you can have a lot of quaternary structure. For example, for virus, or the plant virus I study in my lab, it's the icosahedral virus, and it's composed with 180 subunits. So it's a very large proteins. Okay. So I think it's no, maybe we will do the Menti after the coffee break. So this is a summary of all the lecture I gave you. So we have three classes, three kind of proteins, which depends on their structure and their solubility. So the fibrous proteins like keratin or silk. So it's polypeptide chain, only one type of secondary structure. For example, the keratin is made with alpha helix, and the silk is composed with beta sheet. Form long filaments which are shaped like rod, right, for the keratin, for example. And he has a structural roles, protection and support. You have the globular protein or soluble proteins, which are usually spherical shape, and you have several functions like enzyme. I think you will have a lecture about enzymatic kinetics. Transporter, we will see that when we but transporter is like the albumin, for example. And the last part is the membrane proteins, or it's soluble only in membrane. And we will see that I think the next lecture, we will see the structure of the membrane protein and their roles. Their role is ionic channel, transporter, receptor. So the protein solubility depends on the nature of the amino acid at the surface. So we can have here what we call hydropathic graph plot. You have all the amino acid charge. So if the surface of your proteins have this kind of amino acid at its surface, so charged or polar, it will probably soluble proteins. And when you have hydrophobic and apolar residue at the surface, the protein should probably be membrane proteins. Okay. So this is a summary. You have soluble protein, membrane protein, so in vivo. And when you want to study this protein in vitro, you have to find the optimal conditions to have the best stability of the proteins. So you have to find the temperature and pH of protein stability. You have to use ionic strength, which allows the protein solvatation. So remember, this is the salting in process when you increase the concentration of salt. And for the membrane, you have to mimic the membrane by using detergent. Okay. You have also a modification of the polypeptide chain after its synthesis. So it's called the post-translational modification of the proteins. You can have the cleavage of proteins, the formation of the disulfide bridge, the bending with cofactor to you come from the apoprotein to the holoprotein in the presence of cofactor of metals. And you can have also glycosylation, phosphorylation, and so in this table, I give you the most common post-translational modification. So you have phosphorylation. It means that you give a phosphate group to the proteins. We will see that it will reversible, and it's the phosphorylation. It's a signal to the cell. You have methylation, acylation, fatty acid modification. We will see that it's very important because if you want to have proteins in the membrane, you should have fatty modification. Glycosylation is because it's involved in the cell-cell recognition. And we will see that it will give also, for example, the blood group, and so on. Okay, ubiquitin, you have all this kind. So my last example is insulin. So insulin is a peptide hormone produced by the pancreas. And you know that it regulates the metabolism of carbohydrate and fat by promoting the absorption of glucose from the blood to the muscle. Okay, so it's very important to have this insulin, and usually the diabetic people cannot produce the insulin. So the insulin, after its translation, it's a polypeptide change like this. It's called pre-proinsulin. You will have a signal, and you will have the disulfide called the proinsulin. And after that, we'll have the last modification. It's the cleavage of the chain C. And now you have insulin, which is a peptide which is composed by two chains linked together by disulfide bridge. Okay? So this is, I think, the last slide. Yes. So we can have 10 or 15 minutes for the coffee break. And after, I have a quiz online, and I have to correct the exercise, and it will be done for today. Okay? Except if you have any question.
I think
No?
we can have a break.
Yes. And after, if you have question, I will, no problem. Okay?
Okay.
We can finish. So I told you to prepare this the exercise 16, separation of proteins. Do you do that or not? No?
Yes.
So, yes. Okay, so you can help me to make the correction. So suppose you have a mixture of 5 in this table. Okay, so you have the indication of the molecular mass, the weight of the proteins, and the isoelectric point. Okay, the first question: indicate the order in which the protein will elute from a gel filtration column. starting with the one that elute first. So on the why it can yes. Okay. For the gel filtration, it's also called side exclusion gel. It means that which protein will be eluted first, the bigger one or the smallest one. So the principle of this technique depends on the mass, molecular mass, okay, of the protein. So the first one will be
the bigger one first.
Yes, bigger one, bigger protein will be eluted first. So it's mean C first, and after A, B, E, and D. Okay. Okay. Yes. Second techniques. You load this mixture on the cation exchange column. It means so if you have a cation exchange column, what is the charge of the column? The gel or the resin or the beads of the column will be
Negative.
yes, negatively charged. Okay, so it means that only positively charged will bond to the gel, and the neutral and the negatively charged protein will be eluted. Okay, so to know the charge proteins, you have to remember that the pi, if the pH is lower than the pi, the protein is positively charged. Is the pH value it's equal to the pi, it's neutral. And if it's higher than the pi, it's negatively charged. Okay, so tell me at pH, what is the pH? 4.76, what is the charge of this protein? Olvalbumin, it's positive, negative, quite neutral.
It's negative.
It's negative. Yes. Okay, let's say that the pH is quite similar to the pi. Okay, so it's negative, quite neutral. Okay. The other, myoglobin, the B proteins, it's positive or negative or neutral. The pi is always higher than the pH value. So it's
Positive.
yes, positive. They are all positive. So this the protein will appear in the flow through. It will not bound to the column. It will be the A proteins because it's negatively charged or quite neutral. Okay, and in order to elute those proteins that are immobilized on the column, you then apply a linear salt gradient with chloride sodium concentration gradually increasing from 0 to 1 molar. Indicate the order in which the protein bonds to the column will elute at the salt concentration increase. So when you have the difference between the pI and the pH, this value is low, the protein will elute first. Because it means when the difference between the pH and the pI is low, it means that at the surface, the protein has few charge. But if you have the difference between the pI and the pH, which is high, it means that it's highly charged. You have a lot of charge. So it will be tightly bound to the column. Okay. So when you increase the salt concentration, you will have competition. You will have interaction between the salt and the resin. So the protein can be elute. The less charge have the proteins, the first it will be elute. Okay. Which one will be elute after A? A, we see that without salt can be eluted. So after we have C because it's PI, it's 4.9. And after D, yes. After
B,
B, and the last one, E. Okay. Yes. Okay. Now you repeat the same, but use an ion exchange. It means that this time the resin or the gel is positively charged. Okay. And at pH 8, tell me what is the charge of the protein A, B, C, D, and E. A will be negative or positive?
negative,
Negative. B,
negative,
yes. C,
negative,
yes.
negative,
D, and E,
positive,
yes. So in this ion exchange column, the first one elution will be E, right? Because there's no interaction between the positively charged protein and the positively charged resin. And after, when we increase the concentration of salt, we have which one will be elute after E.
It's A. Oh,
No, The first one will be the one which has a low value between the difference between the pI and the pH. So if the pI is close to the pH, it means that the proteins are not highly charged. Okay, so it cannot have strong interaction. So the one which will be it will be D, the first one. Okay, and after D, after C, and the last one will be A. Do you understand? E, C, D, A, right? No, why you put C before? No. Okay, it's B, D, C, A. Okay. Understood or not? Okay. After that, when we make SDS page interaction, electrophoresis, which one go faster? The smaller one or the bigger protein?
Smaller.
Yes, the smaller protein goes faster or migrate faster. So the order will be, if we have the gel with the SDS, the protein are positively charged or negatively charged? Where for the electrode?
Negatively.
All the protein will be negatively charged. So they will go to the positively charged electrode. Okay, this is the direction of the flow. And you will have the smallest one, which is D, E, after B, A, and the bigger one is C. Okay, and here you have the depot. Okay. And so, the last question, you are interested in further study of cytochrome C based on the result of your experiment device. Which purification is the more accurate?
I think it's the anion exchange column.
Yes. So if you want to purify it from the rest of the mixture, is it's when you elute E and all the other proteins are bound to resin. So the best techniques will be anion exchange column. Sorry for it's difficult to write. Okay. Do you have any question?
No.
No. Let's go to the next one. So, in the next one, we study the denaturation by temperature of lysozyme. So, say no. To do that. Yes, I know. I try to. So you have in the first and second figure the CD spectra of the proteins, okay, from 20 to 95 degrees, okay? So using these two figures, which secondary structure is predominant in this protein, in the egg white lysosome?
Alpha helix.
Yes, because you have two minimums. Two minimum around 200 nanometer. It means you have alpha helix. Let's see if we have something. Yes. Okay. What is the next question? Ah, yes. I try to remove them. Okay. So if we observe this time, the fluorescence spectral change, we use the intrinsic fluorescence. What is the excitation wavelength? The intrinsic fluorescence, it's also the same that the tryptophan fluorescence. So what is the excitation wavelength? We already see that. For proteins, for tryptophan, we excited the proteins at 280 nanometer. Explain the redshift observed of the fluorescence intensity when the temperature increases. If you see, we have a decrease in the fluorescence intensity, but we have also a redshift. It means that the maximum of the fluorescence spectra goes higher, has a higher wavelength. Okay? So I already explained that, but if one of you can explain, I can be sure that you had understood.
Because the excitation normally is belong to the tryptophan, and tryptophan is hydrophobic. So in normal protein, the hydrophobic is located inside.
Yes.
And when we increase the temperature for the proteins of the tryptophan will explode to the outer environment.
Yes.
Yeah, and it changes the polarity of the environment, and
Yes.
it causes a redshift.
Perfect. So I guess everyone had understood. Otherwise, you can explain in Vietnamese. It's more easy. Okay. After that, so we have the redshift because we have a denaturation of the proteins, and because the tryptophan, which is localized inside the proteins in the native form, will be exposed to the water, so its environment will become more and more polar. So that's why we have the redshift. Okay. After the last question, is this denaturation reversible? So you have the green one is the initial spectra at 20 degrees. After the denaturation, you have the blue one. So we observe degrees in the fluorescence intensity and redshift. And when we cool again this solution at 20 degrees, we obtain the red curve. Okay, that means that it's quite reversible, but not totally. Okay, because if it was totally reversible, the red and the green curve should be superposed. Okay, so you have not totally reversible.
Can you explain again one more time why it's not totally?
Because if it was not totally reversible, the red curve and the green curve, which is the fluorescence spectra of the protein at the same temperature, will be the same, right? If you obtain all your protein in the native form. So when you increase the temperature, you have a denaturation, and maybe you can have some proteins which cannot come back to their native form because they have aggregation or something like that. So it's not totally reversible because you have the difference between the red and the green curve. That's all. Okay.
Okay.
And finally, last exercise. So you have a protein. Its mass is 70 kilodalton, which will and SDS page of this protein held a single bond corresponding to this mass. However, in the presence of the reducing agent like 2-mercaptoethanol, you have two bands. So it means that you have the SDS stage so without mercaptoethanol and with mercaptoethanol, you have the negatively charged electrode, the positively charged electrode. So without mercaptoethanol, it means that you have one band at 70 kilodalton, and with the mercaptoethanol, you have two bands. One are 40, the other are 30 kilodalton. Okay? From this data, describe the native proteins in terms of number of subunits, the molecular mass of each subunit, and the stoichiometry of the subunits, and which kind of interaction are existing between the two subunits.
Teacher,
Yeah.
you said in the text is two band of 30,000 Dalton and 20.
Sorry, you're right. I don't know. 13 and 20. Sorry. You are correct. So So what is the composition of these proteins in terms of subunits? How many subunits?
I think 3 subunits.
Okay. So we have 3 subunits.
We have 2 subunits with 20,000
Yes.
and 1 subunit with 30,000.
Yes. If you 17 it to 20 and 1 for 30. It's okay for everyone. So we have 2 subunits with a molecular mass of 20 kilodaltons and 1 subunit with a molecular mass of 30 kilodaltons. Okay. And what kind of interaction do you have between them?
Disulfide bond.
Yes, disulfide bond, correct. So for example, I draw something like this. No, it's very ugly. And you can have disulfide bond. You can also have disulfide bond between them. Disulfide bonds are disrupted by the presence of the mercaptoethanol. Okay, so when you did the SDS experiment in the presence of mercaptoethanol, you will break this bond and you can have the subunits. Okay. Last question: you treat your protein with protease, but in this case it's trypsin, and you run into SDS page. And this time, so in the absence of mercaptoethanol, still show 1 band. So probably at 60 kilodalton, 70 kilodalton, but 4 bands in the presence of 2 mercaptoethanol. How will you explain these results?
I think that the cleavage sites are located in between the sides of the bonding. So so I think that if you use IDS page, you only see one bond, one band, but you the 2 mercaptone ethanol you can you cut the disulfide bond, so you only show the also show the this side of the trypsin.
Yes, probably. Indeed, you probably cut the protein with the trypsin, the cleavage trypsin. Maybe you cut here and here. Okay, it means that if you have, no, maybe not like this. In my case, let's say we have several disulfide bonds, and I can cut here and here, but here also all the part after the digestion is linked together. That's why by the sulfide bond. So that's why we still have one bond. But if we have trypsin digestion and the presence of mercaptoethanol, you will break all the proteins and the disulfide bond, and you will obtain the four band. Sorry. Understood?
Yes.
Okay. Do you have any question about this lecture about protein structure
Teacher,
or the size? Yes.
the first question, what is the stoichiometry of subunit?
Stoichiometry, it means how many subunits you have. So it means here you have 2 subunits of 20 and 1 subunit of 30. This is the stoichiometry.
Okay.
So we just need to describe how many subunit of each
Yes.
type.
Yes,
Oh, okay.
that's all. That's all. Okay. You have to give the different type of subunit with different mass and how many of each subunit you have. So you have 3 subunits, 2 of 20 Dalton, and 1 of 30 kilodalton. Okay.
Yes.
Any other question? No? So we can end for today. We have no class next week, but only the week after. So I think it's 13 December. I don't remember. It's on Monday and Thursday and Wednesday, correct? 13 and 15 of December. Okay? If you have the question, you can ask me by email or or on Monday next week. Okay?
Okay.